AS Biology. Welcome. Are you excited for a fun learning session? Make sure you have your book and a pen before we start. Today you are going to learn about enzymes. Enzymes action. Enzymes are large 3D molecules made by a protein folding into a specific shape. The shape that a protein folds into is dependent upon the order of the amino acids that are present in the protein. Enzymes are biological molecules that speed up chemical reactions. They are biological catalysts that speed up reactions in the body. Enzymes catalyze metabolic reactions, on a cellular level such as respiration for the whole organism such as digestion. Enzymes can build molecules in a process called synthesis. These reactions are known as anabolic reactions. Enzymes also break down molecules in a process called digestion. The reaction is known as a catabolic reaction. Enzymes are globular proteins. There are intracellular enzymes. These are enzymes that work inside cells, and extracellular enzymes, which are enzymes that are secreted by cells and work outside the cells. Enzymes can affect the parts of an organism, for instance by creating DNA, and also by carrying out essential functions inside the body such as respiration, breaking down proteins into amino acids. Enzymes are specific. Each enzyme can only work on specific substrates. Normally they are only able to catalyze one reaction. This is due to the tertiary structure of the enzyme. The tertiary structure is dependent upon the primary structure, the sequence of amino acids. The bonds formed in the primary and secondary structure affect the shape of the tertiary structure, and these together affect the shape of the active site. The active site of the enzyme is the site where the substrate binds. This is affected by the sequence of the amino acids in the primary structure and the bonds formed in the secondary and tertiary structure. Enzymes work by lowering the activation energy of a reaction. The activation energy of a reaction is the minimum energy required for a reaction to occur. Often the energy provided to start a reaction is heat, so enzymes also reactions to occur at lower temperatures and to occur faster. Enzymes lower activation energy by forming enzyme-substrate complexes. This lowers the activation energy by. The enzyme holds the substrate enzymes closer together. This reduces repulsion between the substrates, and so the substrates are more likely to react. The enzyme strains the bonds in the substrate and means that less energy is required to break the bonds in the substrate and break it into two or more smaller molecules. The red line shows the reaction of an uncatalyzed reaction and the blue line shows the reaction of a catalyzed reaction. The blue line has a lower activation energy. When the substrate binds to the active site of the enzyme, and enzyme-substrate complex is formed. How does it work? In synthesis, the two, or more, molecules bind to the active site of the enzyme. The enzyme catalyzes the reaction and the two molecules are bound together. The new molecule is released from the active site. The enzyme is now free to be used in another reaction. How does it work? In digestion, the molecule binds to the active site. The enzyme catalyzes the reaction and the product is released as two or more molecules. How does it work? In digestion, the molecule binds to the active site. The enzyme catalyzes the reaction and the product is released as two or more molecules. Evidence has shown that the lock and key model may not be completely accurate, so a newer model has been devised called the induced model. The induced model suggests that as the substrate enters the active site, that the active site changes shape slightly. This means that the substrate fits more tightly into the active site.